Good evening. It is so nice to have folks joining us. I am joining from the Washington, D.C. suburbs, where fortunately it's gotten cool enough that we do not have active mosquitoes, but it's not so cold like in South Dakota, where Cassie's joining us from, or Colorado, where Rusty's joining us from, or even Alaska, where Liz is joining us from. It's not quite as cold as those places. So greetings. It is an honor people joining us this evening. And if you'll be so kind as to put in the chat box where you are joining us from. And we have a pretty neat evening tonight. We're going to be talking about some of the who can prevent mosquitoes from some of the even natural ways that you can prevent mosquitoes from invading your territory. We have nothing against mosquitoes. Sometimes people will ask me, oh, are you trying to get rid of all mosquitoes? And indeed we are not. Mosquitoes serve a very useful purpose, but it's just a good idea to not have them biting us, around where humans are.
And Rusty Lowe, Dr. Rusty Lowe is the co-host for this webinar tonight, and she's taking over.
Great. Okay, I guess we're getting started. I'm Rusty Lowe. I am the scientist that is working with Globe Mosquito Habitat Mapper, and I would like to just introduce Dory and Janie. She's our campaign coordinator. Cassie Sophine, who is our monitor this time, is the informal science outreach lead. We work with a terrific educator, Liz Burck. And today we have the pleasure of having an absolutely fantastic presentation. And we can maybe make the slides go further up. Oh, we have an announcement that next month, we're going to launch the 2020 Mission Mosquito campaign. Particularly if you didn't participate last year, we're going to use this as a time to sort of regroup and talk a little bit about our goals. We'll talk a little bit about some of the tools, and it's going to be a real kind of basic overview. And as well, we're going to have a guest scientist who's going to be talking about Globe data. So it should be really, really interesting, and that will be on January 9th in the new year. Okay. Today's webinar is really excited. I spent all day waiting for this moment. We are going to have a guest scientist today, Anita Schiller. She is the director of the Biological Control Initiative in Harris County, Precinct 4, Texas. She and her team are really, I would say, probably national leaders in trying to understand how to keep mosquitoes at bay without the use of pesticides and with using the natural environmental relationships that are in our ecosystem. And I can't wait to hear what she has to say about that. And then once we're done with that, we're going to talk about a couple of resources that are available to use in your teaching and outreach, or just to learn more about this on your own. And we'll end up with a spotlight on something that a teacher's been doing with Globe Mission Mosquito. So if we go to the next slide, I'm very happy now to officially introduce our guest scientist, Anita Schiller. I had the pleasure of getting to know Anita Schiller over the past few months. We got in touch with her because she gave an amazing talk at a museum in Texas. And we had some students that were Globe Mission Mosquito Program that were really excited about this topic, and they are the ones that kind of brought us together. And since that time, I've just been really enjoying a lot of the insights that Anita has, and what she's going to tell us today is going to be completely amazing. I don't want to give you her whole bio because it would just take a long time. Just know that she has been working in natural history for quite a long time, and that she an expert on mosquitoes and mosquito management and biological control. So it's my great pleasure to introduce my new friend, Anita. I think you are able to control your own slides. Is that right?
I'm going to attempt. Hi,
She's
everyone.
Very good. Well, she can do so many things. She can probably do that. And I'll be monitoring the chat, so you don't have to, once you're done, we'll get those questions out to you, okay?
All right. Thank you so much. Hello,
Thank
everyone.
you.
I have to tell y'all, it is such an honor that I was invited to present to y'all our favorite topic of using mosquitoes. And I'm just over the moon that I get to talk to everybody tonight and to share this exciting information with y'all. I'd like, as a forewarning, I want to let y'all know that I'm more of a biologist and less involved in technology. So if there's a technological glitch, it's going to be my fault. I'm sure of it, because if there's an on button that it requires, I'm usually the last one to find it. But if it's got things like eyes and skins and wings and what have you, then I can figure it out. So with that being said, I'm going to go ahead and see if I can get this presentation shared. So I heard it's pretty cold where the rest of the team is at. I have to let you know that I'm in short sleeve. And even though I sported a jacket first thing this morning at 6 o'clock, I promptly took we did get into the mid-70s today. And just this evening when I was outside briefly to check on my trees, I did have 2 mosquitoes bite me or attempt to bite me. So mosquito season is always hot here in southeastern Texas. With that being said, a quick note, I am going to turn the video function off on my end because you're not really here to see me, you're really here to see the presentation. All right, so I already mentioned it's kind of warm here where we're at in Texas. And most people are not familiar with where Harris County is. And when we tell them, we usually get blank stares. So most people are familiar with the city of Houston. But city of Houston is really just one of 34 other cities that are part of Harris County. To give you a little bit of knowledge about Harris County itself, is that we have an awful lot of residents that live in ours. It happens to be made up of more folks than the entire state of Kentucky has residing in it. And on a coverage area, Harris County itself, one teeny weeny little county, is larger than the entire state of Rhode Island. So that's quite a bit of landmass. So when it comes to that, we have a lot of mosquitoes to battle because over 4 miles are completely covered, permanently covered by water. The state of Texas has mandated that all counties need to have a panel of 4 commissioners to preside over the county business, and Harris County is no different. And if you noticed in the introduction, you will have seen that my title says I work for Harris County Precinct 4. And the head of our county, Precinct 4 is Commissioner Jack Cagle. Now, he is the spearhead and the leadership of our organization, and he pretty much lets me do what I need to do in order to come up with novel and better ideas on how to deal with these mosquitoes. So I already mentioned, we're pretty warm here and we got an awful lot of water. So all year long, we have a biting pressure of mosquitoes, and those are made up of 56 different taxa. All year long, unless there's a hurricane blowing them away, we have mosquitoes biting us. But this presentation is not really about all the mosquitoes, but I have to give a little bit of information about them. And again, this presentation is built in the US. So I saw that there were several participants joining us from other parts of the world. So you will have a different diversity of subfamilies in Europe. For the most part, they'll be the same. So we have here in the US, we have 3 main subfamilies, or main categories of mosquitoes, family Calicidae. And those are made up of Toxovincinidae, with just 1 genus represented here. Anophelines, or Anophelinae, that is made up of the animal or the mosquito that can transmit malaria. And then there are 2 other genera part of that. And then the largest by far is Calycinae, that contains 30 different genera with roughly 2,000 known or described species. And the majority of those are also medically, or a large number of those are medically important. You can see on this particular slide that I have grouped them. Toxorhynchinae on the left, Anopheline in the middle, and then the large Calycinae on the right. The red arrows indicate the individual markings that set them apart, besides, of course, genetics. So on the left, Toxorhynchinae, the white dots on the black background are actually eggs. The next picture with the arrow is a mosquito larva feeding on another mosquito larva. The pupa shape is pretty much consistent across all the mosquito subfamilies. The adult is fully obligate autogenous, meaning she and he, both male and females, are sugar feeders, solely sugar feeders. They will never take a blood meal. On the head structure, the proboscis itself is curved, and in fact, it is only able to sip nectar from flowers. Anophelines, the eggs are laid on the water surface. They often clump together, but they have little allow them to float and scooch across the water. The larvae of Anophelines lay perpendicular to the water surface. They lack a siphon. And so you see that is resting up against the water surface so that it can breathe. Pupa, once again, looks very similar to all the rest. But then in the feeding, host feeding portion, blood feeding portion, the Anopheles will often sit head down and abdomen up, almost upside down. And that's quite a characteristic that is useful for the ID. Hopefully nobody lets the mosquito bite them long enough to see that. And then if you look on the bottom, the head structure, you will see are just as long as the proboscis, and so it looks like they sport a fork on their head. Aedes eggs are laid above the water line. They are dry. They often dry. They're laid singly. The larvae usually sport a semi-short siphon. Pupae, once again, look similar across the board. And they're quite varied in their ovipositioning habitat. Culex lay rafts that are clumped together. They have to lay them on the water surface. Should the Culex raft leave the water, it will dry up and the progeny will be doomed. Culex siphons in the larvae are usually long and slender. That's, the emphasis here is on the word usually. Pupae look similar. And the mosquito adult is often a dull brown color, pretty nondescript. And I'm going to leave it at that because this presentation is really about one particular kind and not so much a full mosquito primer. I could go on and on what makes all these mosquitoes unique. With the last one over here on the right, the Mansonia and Coccolatidia are actually some of my favorites because they are so bizarre. But perhaps we leave that for another presentation. All right, so I am really consider myself to be a cheerleader because they're not all bad. And I'm often asked, well, geez, are there any good mosquitoes? And can't we just get rid of them all? And my usual answer is, an eye roll because we're not privy to all the information. We're trying to find out the information, but we don't really have it all. We don't really know why animals are put on this earth or why they've evolved to fill a certain role or a niche in the environment. So one good illustration, though, that I have that is near and dear to my heart is what good are mosquitoes? Well, if you happen to like wild things that are ephemeral, and unique, you might actually enjoy seeing wild and native orchids. And in this particular case, this blunt leaf orchid, or also called the small bog orchid, it is required to be pollinated by, you guessed it, a mosquito. So if we were to remove Aedes communis from the environment, if we could snap our finger and just take all the Aedes communis off the earth, we would also remove this orchid. So for biodiversity, mosquitoes in general do play a role, and they are important. But once again, this presentation really is about mosquito assassins and not about all the other mosquitoes, and I'm merely a cheerleader. I wanted to throw that out there so that everybody has a better idea, or can finally lay that question to rest. What good are mosquitoes? All right, so let's go ahead and dive into our mosquito assassin. We have, as I mentioned, a couple native ones in the US. And so we'll discuss this a little bit more in detail. We will go over the classification, where it fits within taxonomy and the classification of animals, where you might find it, its life cycle, its odd oviposition behavior that's different from all the other mosquitoes, the juvenile development that's also pretty unique, or pretty different, and then the habitat overlap and why we might be able to use it as a biocontrol agent. All right, so the taxonomy. I'm sure y'all are familiar that they are arthropods. They belong in the group of insects, and they are true flies, so they only have 2 wings and a set of halteres. They're part of the mosquitoes in midges groups and the family Calicidae, or mosquitoes specifically. Within the mosquitoes, the tribe or subfamily Toxorynchinidae is made up of 4 genera, and those have 90 different taxa assigned to them. By far the largest is Toxorynchides, with currently 51 described species, although I know that there are several undescribed species that a colleague of mine and mentor of mine has in a drawer that are yet to be described to science from South America. However, the US has home to 2 species, Toxorynchides rutilus, which is what this presentation is mostly focused on, and also Toxorynchides moctezuma. Here's the distribution for the United States. You see the large black area is Toxorynchides rutilus. There's also a small area in Florida that's also Toxorynchides rutilus, although that's a subspecies, Rutilus rutilus. And then the most ubiquitous one is rutilus. And those really are just differentiated by the males. The female's hind tarsi having a white ring versus a white interrupted ring. And then Toxorynchides moctezuma, there are a couple of records for it in the far, far south desert in Arizona. And the very far tip of Texas, Toxorynchides moctezuma reaches in there. Moctezuma can be found further south, all throughout Mexico, or I say all, I mean in several parts of Mexico. And generally they're very closely related to Toxorynchides rutilus, whereas rutilus requires a more temperate and higher humidity in its habitat. Toxorynchides moctezuma prefers drier areas. But as far as the rest of the world goes, you might find Toxorynchides genus in the subtropics across the globe. And here we have a screenshot of an iNaturalist map where mosquito assassins, different Toxorynchides species have been observed, and you will notice that the majority are in the tropics, and then a hot blob over the US. But where in the environment might you find Toxorynchides? In general, they share similar habitat preferences, and they are indeed a native predator or a predator of rain-filled vessels. They most often preserve prefer natural cavities that are filled with rainwater, that are ephemeral. Sometimes those will dry up. And in general, those containers, as we call them, just lack predators, other predators. Like in semi-permanent waters or even permanent waters, you will have dragonflies and fish and even big tadpoles that might eat them. But in general, these ephemeral rain-filled containers lack those predators, whereas Toxorhynchides will then become the top predator. I have a few pictures up here on the types of containers where you might encounter Toxorhynchides, and incidentally, of course, other mosquitoes or biting mosquitoes as well. They do prefer brush and sylvan-rich or rich in greenery areas that can be landscaped, so artificial, but it also can be just natural environment as long as it's rich in plant material with cover and shade and sunlight, and also flowering plants from which the tox can feed from. That's where you might find them. And for the US, here in Southeast Texas, they will actually fly and be active as adults from late spring on all the way through fall. These are a few images of where you will find them, or where I have actually found them, all the way from the jungle in the Yucatan, limestone solution holes, down in tree cavities, big tires, tractor tire towers and tire dumps are just a few examples of where you might find them. So let's go into the life cycle of our mosquito assassin or Toxorhynchides, because they are so unique. They are a holometabolous insect, so they have a complete life cycle that starts out as an egg, a larva stage that has 4 different molds or stages, a pupa stage during which metamorphosis occurs, which is similar to the pupa stage in a caterpillar, where the caterpillar breaks down and then gets wings and emerges as a butterfly moth. And then, of course, we have the adult that comes off the water. In general, these animals that start out in the water but then are flying adults, they could technically remain in the water, but since they are in ephemeral containers, they would stand the risk of drying up. And of course, they will need to find a mate. And should they remain in the water where their siblings are, inbreeding would of course be a problem. So it behooves the animal to grow wings and be able to disperse and find an unrelated mate. Let's see, duration. For toxins laid onto the water, and these numbers are for my local area. So in Southeast Texas, the eggs will remain in egg stage and embryonate in about 2 days. After that, the larval stage can complete as quickly as 13 days, or it can take a few months if it draws into the winter, during which the 4th instar will diapause. Pupa stage can take anywhere from 4 days to 8 days, roughly, but in the summer it'll complete in about 4 days. And then the adult, if it's a male, it'll pass on its genetic material and die within a week. Females can last a couple of months if they don't get eaten. So in our cages, production cages, our females can actually last a good 2 to 3 months. But in the wild, we expect them to fall prey within 15, 16 days or so. The large picture, the background picture, depicts a female Toxorhynchides rutilus, and you can see its beautifully adorned body that are metallic scales. Those are golden scales, purples and blues. And then the bottom right picture, the smaller one, is the male, and you can see the fluffy antennae that make it very obvious that it's a male. One of the unique characters or oddities of Toxorhynchides biology is that the eggs are laid individually, and the eggs are hydrophobic. They scooch across the water, And the embryonation is temperature dependent. As I mentioned, they can the embryos can eclose in about 2 days, although they could take a little longer if the temperature drops. But what makes their ovipositioning unique is that they will seldom, if ever, the females will seldom, if ever, land on the water, because there may be an occasional spider waiting for them. So it behooves them to stay clear of the water, and that gives them this very unique oviposition behavior that looks like this. And I hope that the video comes through on y'all's end. The female on the right is actually depositing an egg. I'm not sure that you saw it. I hope you did. You saw that the egg was deposited during the flight, and the Toxorhynchides in general has this looping oviposition flight during which the egg is simply tossed from the abdomen. And here's a better close-up of what the eggs might look like up against the leaf. That's in a natural environment. The eggs do have a slight charge to them, and so they will clump. But as I mentioned, they're hydrophobic, and if rain happens to occur in an open vessel, excuse me, those eggs stand the risk of getting bounced right out of the container. But they will clump together through the charge, and that's kind of a built-in adaptation, because these animals are actually so hungry and so aggressive that they're also cannibalistic. And so you will have the older hatch or the older progeny actually cannibalize on the younger ones. As the younger embryos eclose from the eggs, the older ones are waiting right there to eat them. That might seem cruel, but in the end, only 1 or 2 will survive, but we can be sure that those are big, brawny, robust animals. As I mentioned, the larvae have 4 instar separate molds. They're highly aggressive, so that makes them really good as a biocontrol, but also limiting. They have a variable development time, which can be as few as 13, 14 days. It can take a couple of months, depending on the season. And of course, the development is triggered and dependent on temperature. If it gets really cold, they will slow down. The photoperiod will tell them mostly whether it's time to speed up or slow down, and of course, how much food is available. Here's a Toxorhynchides larva feeding on some aquatic nematodes. And just an FYI, that video was real time. It was not sped up. So that's how fast a tox larva, this is an L2, will actually consume a mosquito prey, or in this case, a small nematode, and be reset and ready to eat more immediately thereafter. I've already mentioned a couple of the distinguishing features. This is just a refresher here. The proboscis is downward curved for feeding on nectar. The picture on the left depicts a female. I'm not sure if this will come through on everybody's screen, but if you can look at her abdomen, you will actually see eggs already developing inside her abdomen. And the picture on the right is a male with his plumose antennae, and his maxillary palps are almost as large or as long as his proboscis, and they're curved to the side. The old name for a mosquito assassin is actually elephant mosquito, and the picture on the left of the female will actually show you why they were once called elephant mosquitoes, because they do look like they're sporting an elephant trunk. So the female, as I mentioned already, is obligate autogenous, which is a big fancy jargony word that means she is obligated to never take a blood meal. She cannot take a blood meal. Even if she tried, she couldn't take a blood meal. And because she takes in so much food during her larval development, i.e. other organisms that are full of fat, lipids, nutrients, what have you. She doesn't need that blood meal. Like pest mosquitoes that feed on algae and detritus, they lack those nutrients in the water, so they have to have that blood meal afterwards for those nutrients to develop eggs. Toxorhynchides doesn't need it. She takes in all the nutrients that she needs during her larval development. The male, well, he pollinates flowers, does his deed, and then dies. So you may recall the title of the slide deck being when the SWAT team, the mosquito SWOT analysis, excuse me. So I found this little cartoon and I thought it was absolutely hilarious. The mosquito youngster playing video games or watching TV and wanting to meet the SWAT team. So here's our mosquito SWAT team. So let's go ahead and do the analysis. I hope I can get through this quick enough for everyone. The SWOT analysis is actually a business analysis tool. When we look at an opportunity, we look at all its strengths, its potential weaknesses, its advantages or opportunities, and the threats. So for Toxorhynchides to use as a biocontrol agent, for the U.S., Toxorhynchides is native. They're indigenous, they're endemic to the areas, they're benign and beneficial. There are no regulations about using them in the U.S., and there are no known environmental risks. If we supplement existing populations with more, that really just translates in less mosquitoes, and that's a good thing. They are absolutely adapted to the local environmental conditions, meaning they can deal with our droughts, our hurricanes, our floods, our tornadoes, our brutal heat in the summer and everything else, and the occasional winter that we have. They can overcome prey shortages, which I'm not sure that is ever the case really, but should they ever find themselves in a vessel or container where mosquitoes can't find them, they will overcome that via starvation. Toxorhynchides retilis can starve, especially in its later instar, for weeks. They can overcome a summer drought during a quiescence or a pause of development, and they do something similar called a diapause in winter, where they absolutely cease development and then come out of diapause as photoperiod lengthens and temperatures warm up. Their weakness, of course, is that they're absolutely aggressive feeders, so aggressive that they can be cannibalistic, which is, well, good in one way, but bad when you're trying to supplement a population. They also don't fly a very large distance, which is a hindrance as far as development, human development is concerned. In Harris County, at least, and surrounding counties, there's population boom and development boom. So there's a lot of habitat development and destruction going on. So the Toxorhynchides boundaries are getting pushed further and further back. And so they don't really have a quick natural dispersal to migrate back into areas to where they were removed. So we say that their dispersal distance is relatively short, so we help them out with intentional releases. They do have a limited usefulness. So if we have mosquitoes coming out of storm drains, breeding in storm drains, Toxorhynchides, for instance, is not the right biocontrol method or agent to use there. So a careful matching of target and biocontrol agent is necessary, and that limits them for a certain extent. And then the real problem is that you can't order Toxorhynchides or Mosquito Assassins out of a catalog or website or anything. And if you could, you can't warehouse them. So the production for their use is still required to be in-house, and that can be quite laborious, dedicated resources to do that. However, their advantage for using is that it's an absolute quote-unquote green strategy. The emotional response of most folks is that they're excited, and the strategy is widely accepted, unlike some newer technologies that are coming onto the market. They are an absolute valuable tool in an IMM or integrated mosquito management program. And they are an absolute superb ambassador for outreach, for teaching folks about what causes mosquitoes, the mosquito control outreach messaging. The threats, of course, is that because they're an insect, they are susceptible to mosquito and other pesticides. And as already mentioned, clear cutting and habitat destruction is a true threat to them. So with that, I'd like to show y'all how we in our laboratory at the Harris County Precinct for Biocontrol Initiative are overcoming the problem of not being able to order this agent that we would like to use more widely. And here is how. This is one of my staff members. Her name is Mary. She has co-developed the rearing protocol that we are now using. And I am originally from Europe, so I'm a metric person, and my brain uses metrics. So with that, I share my metrics for, we calculate things in our laboratory by the hundreds or by the thousands. And in this particular presentation, I'm using the numbers for a cage, one single cage of Toxorhynchitis rutilus that contains 1,000 mixed gender adults. In order for you to maintain a 1,000 strong cage, you have to calculate your resources and your time commitment to replace, on average, 800 to roughly 900 adults every month. And that breaks down to keeping roughly 300 eggs back each and every week. That turns into roughly 200 hatchlings, and those will, God willing, turn into 180 adults. So that will give us our estimated 800 to 900 adults that we need to use to replace the loss that's caused by old age. By the way, if anybody is interested in diving into Toxorhynchitis rearing, we have gone ahead and published our protocols with alternative methods for rearing tox in the laboratory for large productions. The alternative methods will keep you, or will teach you how to produce tox without having to raise prey mosquitoes for their food. So there are other ways to raise them other than feeding them mosquitoes. And those are in this link, in this particular slide. The link will take you straight to our paper on the rearing protocol. But why would you want to go ahead and do that? Because if you wish to use Toxorhynchitis as a biocontrol option, you need to have the product available. And by having one cage of 1,000 animals, and here in this slide you see one of our production cages that are suspended from the ceiling. Those are custom cages that BioQuip suppliers make for us. One of those cages has the potential to give us per year roughly, and this is a conservative number, about 200,000 eggs. That breaks down to 3,000 to 5,000 eggs per week, or 17,000 roughly eggs per month. There is a seasonal fluctuation. So in April, May, and June, you will get probably close to 6,000 or even more per week. And in August and early September, that number will probably drop down to 3,000, even if you keep that number of 1,000 adults steady. In the cage, our domesticated line is not produced like a pet. We don't baby them anymore. We really produce eggs, and therefore we have taken away all the cage furnitures, the pretty things that might give an enriched environment. Our production animals are domesticated. We have colonies that are over 150 generations old. They are not showing any signs of slowing down. But in this particular video here, you see once again how the female lays eggs, and the white clump on the bottom of this 12-ounce stadium cup is a clump of eggs that's probably roughly 1,800 to 2,000 eggs strong. There, I hope y'all saw her depositing that egg. I hope that came through. That makes them so unique and so bizarre at the same time. All right, so now why would we even want to go through that trouble to keep those cages of 1,000 to produce 200,000 eggs a year? Well, here in Harris County, Southeast Texas, we do have a problem with biting mosquitoes that also overlap in the same type of containers. They are yellow fever mosquitoes, Aedes aegypti, and Aedes albopictus, the Asian tiger mosquito, both of which are proving to be quite burdensome during daylight hours. They are opportunistic feeders, and they are pretty domesticated, if not paradomestic animals. Where there are people in Harris County, you will find these two Aedes. Their development is quite rapid, and it's not rain dependent. So even during a drought, you will have Aedes aegypti and albopictus populations quite burdensome, where the public health department may get calls for nuisance biting, requests for sprays. Just because they are domestic around people, they will exploit our backyards and the vessels that we have in our backyards that often look like flower pots, saucers, doggy bowls, water bowls, small swimming pools, kiddie pools, and even kid toys like the little tykes and Tonka trucks will have enough water in them for both of these Aedes to breed in. And of course, the problem with them is that they're competent vectors for arboviruses or arthropod-borne viruses that have recently been in the news, have made the scares. I'm sure everybody has heard of Zika, also chikungunya, dengue, and to some extent even West Nile virus. And then on the veterinary side, they're also vectors for dog heartworm. But those are not the only breeders that, mosquitoes that bite in Harris County. Actually, our primary West Nile virus vector is the southern house mosquito, or Culex quincunfasciatus, which is part of the Culex pipiens complex. And they're a little different where they will lay their eggs. They're a little bit more opportunistic in their egg-laying habitat. Not only will they lay eggs in stagnant waters that are rich in organic matter, but they will also breed in backyard containers. As I mentioned, there are in Harris County the primary vector of West Nile virus, and what complicates things is they really prefer to take blood meals from birds. They're avian feeders. Every now and then, though, they will find themselves in the vicinity of people and then bite people. Usually, they will only take one blood meal, though, and you might say, well, if they only take one blood meal, lay all the eggs, and then die, how can they possibly transmit a virus from a bird to a human if they only take one blood meal? Well, in the case of quinks, as we call them, the virus itself, West Nile virus, can actually transcend through the ovaries. And so a female that has taken a blood meal from a bird that was infected with West Nile virus that lays eggs can actually pass that virus on into her eggs. And the ensuing embryos or progenies, mosquito larvae, are now hot or already infected with the virus. So when they come off the water, they're already sick. And should those find themselves in the vicinity of humans, but not birds, they may actually take a human blood meal. Now, the good thing is that most folks' immune system can combat West Nile virus if they're healthy and young and able, although the very young and the older folks or immunosuppressed folks are at higher risk. But most folks are asymptomatic, and of those folks that do have a higher risk, usually only about 1% or even less than 1% will show symptoms or will actually become ill. So here's just quickly a couple of charts, and my disclaimer is that this is unpublished data. This is not yet in our next publication that is being prepared. This is meant solely for illustration as to what you might expect to see in the wild using Toxarincades as a biocontrol agent. We have run a whole year's worth of studies at a site that is in the glass house at the Houston Museum of Natural Science. Cockrell Butterfly Center allowed us to use their giant conservatory as our little laboratory this year. And what you see on the graph are actually larval numbers of overlapping larvae of Toxarincades larvae that were found in bromeliads. And here you see me in the bottom right picture peering into a bromeliad and taking samples. Toxarincades larvae found in those bromeliads overlapping with Aedes albopictus that have found their way into the Cockrell Butterfly Center. The dark blue highlighted area of the dates in the bottom axis actually shows you the release dates where we released adult Toxarincades, and then the progeny was found in the bromeliads. So we did not put larvae into the containers. The adult Toxarincades laid the eggs into those containers. The pink line is Aedes albopictus larvae, and you can see there's a little bit of lag from the time that we released Toxarincades adults into the environment until they finally had a markable impact. That is a good 2 weeks of lag time, and that is expected if you think about Toxarincades taking a good week or so before larvae are large enough to really eat everything around them. They did have a marked impact that suppressed Aedes albopictus numbers inside that Cockrell Butterfly Center throughout the releases and even thereafter. On the next slide, you see a comparable chart, although that is field data. Once again, it is unpublished and meant solely for illustration. This particular chart depicts adult Aedes albopictus that were caught in field sites. You see on the top right image a aerial shot of the field site with 3 pins. Those are the location of the traps. The bottom right actually shows the trap setup. Those are laboratory grade traps that Biogens in Regensburg, Germany produces. Those are made specifically for Aedes surveillance, and that red cooler above the trap setup releases carbon dioxide. Those 3 traps were monitored weekly, and the chart actually shows the impact that, once again, adult releases had on the adult numbers that were found in the traps. So you can see a marked reduction in Aedes albopictus across those 3 traps. You also notice at the end of releases, you see a resurgence of Aedes albopictus, which doesn't look good on this chart. However, in follow-up research, we found that if you look on the top right picture on that aerial view, there is a wastewater treatment facility. They actually had a incident that just coincided at the end of our releases, where there was an incident at that facility, and they actually produced an ungodly amount of mosquitoes quite by accident. Those numbers are reflected on that chart. We were not anticipating that incident to occur. But anyway, you do see that lower numbers of Aedes albopictus are shown on the graph right on top of when we released adult tox in that same area. All right, so how might you use Toxarincades? Can you just start up a production company? Can you build a facility and just release thousands and thousands and thousands of Toxarincades into the environment and say, all right, we're doing a mosquito control program? Well, you could say that, but it would be foolhardy. You probably wouldn't want to do that. Their best use is as part of an integrated management program, where first of all, the program is set up like an integrated pest management program where vigilance, surveillance, and monitoring go hand in hand. If you don't know where the problem is, you really can't fix it. So monitoring is always important. And on an individual basis, that can be as little as walking your yard every week and making sure that there are no places where container breeding mosquitoes can take up refuge or can lay eggs in. And of course, in a larger place like a district, control district, they would have inspectors go out and monitor different areas in their district. This one often gets overlooked in an integrated mosquito management. Biodiversity, conservation, and promoting beneficial organisms is a new concept. Doesn't always get incorporated because frankly, it's a tough one. Biocontrol itself is its own science, it's its own niche, and in order to use biocontrols with whole organisms, not only do you have to understand your target organism that you're trying to kill, but you also have to understand the biocontrol organism's biology, so you have to know two things to deal with one. And so that's a tough one. A lot of districts are reluctant to incorporate it. However, in a common sense approach, it behooves us to promote biodiversity, to let beneficial organisms do some of the work for us. Source reduction is an easy one to remember. The less water, the less mosquitoes. Larval suppression in the areas where we can't suppress them manually, where we can't tip out those plant saucers or drain those pools. Perhaps we can suppress the larvae that we otherwise can't deal with. And the larval suppression in an IMM program does not necessarily mean just mechanical removal. It can be, take on the form of a larvicide or even microbials. And then the very last step really should be adulticiding. Even though my program focuses on biological controls, fighting nature with nature, in a true integrated mosquito management, we cannot overlook the need for pesticides, although they should never be the first step. Those should be the last. Whenever there is virus in the environment, which goes in with the monitoring part, or whenever emergency situations arise. In our case, it's hurricanes or floods. When those types of catastrophes occur, adulticiding is necessary. In Harris County, we are fortunate enough to have a very well-run public health program and a very large mosquito and vector control program that uses monitoring their adult mosquitoes as their first step. They do look for viruses in the mosquito population, and only when the collected mosquito pools, those samples come up hot or infected with the viruses thereafter, will they send their spray trucks out. Because too much pesticides can backfire on us. Insecticide resistance is a true problem, and it's a true threat to actual public health. Because if we overuse pesticides, then in the end, when we really need them, in the case of an epidemic or even a pandemic, if the insects are resistant to the insecticide, then we won't have a weapon really to combat them. So that's why I'm a proponent and advocate to use adulticides judiciously and as a last resort and only when warranted. So for that, I know I went a little bit over, but I hope I gave you all a good primer. They are something really to look at, why they're important, what makes them cool and unique. And with that, I'd go ahead and open up the Q&A.
Well, that was really fantastic. And I also want to thank you, Anita, for spending some time to talk about the integrated pest management approach and describing all the reasons why it's really important to take that kind of approach, which of course your Mosquito Assassins are part of, because of the problem of resistance to pesticides and the fact that the pesticides are really decreasing our biodiversity. So that's really, really fantastic. So if people have any questions, go ahead and put it in the Q&A. And Anita will be with us until the end. We still have about 10 minutes of material we'd like to cover, if it's okay with everybody. Do you want to cue up the next slides, Cassie? Great, thank you so much. So I just wanted to let you know that we're going to highlight 2 resources today that might be useful for you when you are doing your working with students or doing outreach or trying to learn on your own about mosquitoes. And this is actually the connection that we had with Anita. We had a student in Texas. His name is Parker. And he was doing a study he was doing an experiment looking at using botanicals as ways to attract mosquitoes. And he found one of these Toxyarachnides rutilis in his trap. And then because of that, we connected him with Anita, and that's how we got to know her. So you don't have to read this now, but just look down there. There's a URL. Every month we have 1 or 2 postings to the Science Cafe. We have scientists that are full-time practitioners, as well as scientists that are citizen scientists that contribute to this blog. And if you have something you'd like to share, please go ahead and let us know, and we would love to highlight your research. So please take a look at that, because there's a lot of great stuff in there. Now, we're going to quickly move on. I just want to show you this wonderful picture that Anita sent. Anita, this is your team, right?
Yes.
Yeah, and you want to introduce your people there. There's you, and then there's Adam.
There's Adam, my wonderful assistant who always answers all my technical questions with a smile. Next to him is Mary. She is the rearing specialist. She is in charge of pretty much producing the animals that we quote unquote play with, that we do the research with, and that we release. And her wonderful sidekick is Ari. She's on the right. And we're actually missing another person, but Ari, she is a whiz in rearing Toxyarachnides larvae. She is in tune with them. She can look at them and say, oh, this tray needs some more worms, and she is totally in tune.
Well, I really love this picture because Anita is the lead scientist, but none of this can happen without a great team of people that are dedicated to the mission. So I love that. And they also like to have fun. And they are using here some mosquito tellers that Liz is now going to talk to you about briefly, because you can use these also when you're teaching about mosquitoes.
Right. Because you probably recognize these, right? From a long time ago, maybe, but they probably bring back a few memories. And for us too. So we jumped on this idea of using these as a tool, as an educational tool, and they have been very popular. So I'm sure maybe you remember how to use these. They're the little fortune teller things. And so you choose an opponent like Cassie. Are you ready? On the outside of each of these are the 4 life cycle stages of the mosquito. So the eggs, the pupa, the larvae, and the adult. And so the first thing you do, of course, is you pick one of the outside ones. So Cassie, what's it going to be?
Culex.
Well,
no. I'm sorry. You always have one of those people you play with.
It's still fun. Go right ahead, Kathy. No, one of the life cycle.
Oh, I'm sorry. Pupa.
Pupa. And so we encourage a little bit of spelling practice here as well. So we ask that you have the person operating the teller spell out the word as they open and close. So just P-U-P-A. It opens then to different genera of the larvae, showing the larvae in the picture, but then having the genus name written by that. So which one are you going to choose? Let me guess. Go ahead.
Culex.
The Culex. And again, it has a number there. You can use that number, or you could just, again, spell it out. So C-U-L-E-X. And at this point, you could continue, or you could have the person in making that choice open up one of these. And so I would open it up, and then I would say, oh, this is your question, Cassie. Emptying a water container reduces the number of mosquitoes in the area. So is that true or false?
True.
That is true. And that, my dear friend, is worth 8 points. So you have just collected 8 points, and then you continue to play back and forth. So it is a game. However, we also recognize and want to point out some of the value of doing this. Not only is it fun, and not only is it a game, so it's a contest, and kids love those, adults love those, but it is an engaging activity. And that's one of the 5 E's, right? One of the first ones. So it's a good way to engage people in the study of and the discussion of mosquitoes. It's a hands-on activity, and so they're actively using it and actively engaged. It sparks an interest in mosquitoes because you have to understand the question to be able to answer the question. It does ask the students questions, and the teacher isn't even involved. And so it's a student working with another student on finding information. And it encourages social interaction, which is also a very important component of any informal education idea. So we've developed 5 of these. And so they center on different topics related to mosquitoes. So in this diagram, in this slide, you see we have the green one, which is the mosquito biology teller, which means every one of those questions will have something to do with the biology of mosquitoes. The one on the right-hand side, the reddish one, is the prevention and protection. Again, the title is right underneath the teller itself, prevention and protection. We have 3 other ones. In the next slide, you'll see that we have one for the malaria, one on the left, in the middle is the Zika and dengue, and then on the right-hand side is the West Nile virus. And so again, those are the topic areas. You will need to print these off, and then you'll have to do a little cutting and folding. The directions are included to do that. So they are pretty simple to make. In fact, the kids also enjoy doing those. And another thing that we've decided to use and thought this would be also a really good educational part of this is to have the kids think of the questions that they would put into a teller. What are you curious about? And then can you find the answers, kind of a thing. And so the next slide will show you, we have also produced a blank one. And so you can just have the kids make their own mosquito teller. And so that's also a really cool idea, and kids love that one as well. I've done this with my grandkids. They loved it. I still am finding them all over my house. We have done it with at different meetings of informal groups, and we presented these at conferences, and they are very popular. They're snatched up, and they would be a perfect tool for doing any kind of informal education or outreach. And they are on the Mission Mosquito webpage under resources. So have a look and use them at your next event. I guarantee they're going to be very popular.
Thanks, Liz. And I just wanted to also point out that they are available in color, but there's also a grayscale version in case you're color ink challenged, or that can be expensive.
Expensive, right?
Yep, and then there is the do-it-yourself one on.
All right, so I wanted to just finish things off by letting people know that we do appreciate and keep track of the many fantastic people who submit mosquito habitat mapper data. And as of during the month of November, we had a total of 544 new observations, and you can see that means that since May 27th of 2017 to now, we have had a total of 19,131 observations. Inez, I know you are joining us this evening. You are on our honor roll of Globe members who have submitted the most data, mosquito habitat mapper data, during the month of November. So we salute you, and we salute our other folks there. You'll see that Senegal is extremely active when it comes to taking and submitting the data. And for all of you who are out there, even if you do not have active mosquitoes right now, still take your your smartphone or your iPad, go out there with your mosquito habitat mapper and document the potential places where mosquitoes could be laying their eggs, because that's super important to become aware of and alert to the potential mosquito habitat, location. So anywhere that you have standing water. It's also a really good practice to make a mosquito trap. And for those of you who are in areas where it's not active mosquito season, you can even just use empty cans or yogurt containers. You don't have to do the whole, soda bottles, because you're not anticipating that you'll have active mosquitoes right now. And you can be just keeping track of, when and where we're going to start to see active mosquito season. So we salute all of these folks who are submitting their data regularly. Next slide, please. We also wanted to shine a spotlight on we want to shine spotlights every month on formal and informal educators who are doing amazing things with the people with whom they work. So in this case, we are honoring and spotlighting Mercy Molina. She is a high school teacher with the Prince George's County Public School System. She teaches at the International School at Largo, which is a school for students who are English speakers of other languages, most of whom have only been in the United States for 4 years or less. And she decided that she really wanted to have some authentic investigations for her students to engage in that they felt connected to. We talked about how to have them use their smartphones and cell phone technology to go out and collect data. And I came in and interacted with her students a few times, and it turned out that they were really, really fascinated with learning more about and studying mosquitoes. One of them had had dengue, another had had chikungunya, and they were just fascinated with this whole idea of studying mosquitoes. Next slide, please. The only problem, though, was that they don't have active mosquitoes during the time in which Mercy Molina has her students. They started in September, but by the time they really got going with class, it was October, and they'll be finished in January. So we talked about what are some of the things they were interested in, and what were some of the authentic ways in which they could learn about mosquitoes and take into consideration their local environment and, kind of collect data. So they decided that they wanted to learn about a couple of different things, and they broke up into 5 small groups in their classroom setting, and each group selected a different question. So the first group decided they're going to learn about what species and genera of mosquitoes are in their state and their county. Another group is going to work on what kinds of mosquito-transmitted diseases have been reported in their county and their state. What is the active mosquito season? Another group is going to look at what is the length of the active mosquito season and how is that changing over the past 20 years? Is it expected to change in the future? And then finally, what are the best practices for protecting and preventing being bitten by mosquitoes, which was exactly what we were learning about today from Anita, as far as what is being done potentially in Texas. So each of these groups are going to be collecting atmospheric data. They actually started that way back in September, collecting atmospheric data. And then they're also doing a lot of research to see what is the information for Prince George's County and also for the state of Maryland that would answer those questions. And they'll be using mosquito traps in their homes and then also outside of their homes, and then pulling all of this data together and writing international virtual science symposium reports and submitting them to the GLOBE program so that they can share what they're learning about mosquitoes in their environment and the potential for mosquito transmitted disease with the larger community. So we salute you, Mercy Molina. We're happy to have you joining us this evening. You and your students are doing phenomenal work, and we invite all of you to consider joining us and learning more about, how you and your students, even if you don't go out and are interacting with active mosquitoes. I saw recently that we had a teacher who works in a community where they're very concerned, and rightly so, about EEE and the transmission of that. So they don't want their students interacting with, active mosquitoes. But there are many ways in which you can go and collect data and do research, and then look at some of the data that you're collecting, perhaps through the GLOW protocols, and then make some cause and effect relationships, and also work on better understanding what are some other things that you would need to know to be able to answer your question more thoroughly. So thank you very much, Mercy Molina, and your students. What you're seeing in these pictures are, I brought in some live mosquito larvae that I got from the University of Maryland entomology lab. And the students were able to correctly identify Aedes aegypti and Culex mosquitoes using the larvae that we got from the lab. Of course, they're not reporting that as GLOBE data because we did get it from the lab, but it's giving them the opportunity to be able to use the cell phone magnifiers and go through the dichotomous key and have the enjoyment of working with larvae, even though they, at the current time, don't have larvae in their environment. And you guys have stayed very, very late this evening, or who knows what time it is and where you are. It may be very, very early in the morning. But we thank you so much for joining us. We hope that you will consider working with us if you're going to do an IVSS project, or you're interested perhaps in learning more about possibly doing science investigations with your students. I put a link up there where I'm collecting some data on people who are potentially interested, and I've started a separate base camp for those people where we're sharing resources and ideas and investigating questions. So we really appreciate having you with us. And thank you, Anita, very, very much. Did anyone have questions? And somebody may need to mute themselves, but does anyone have a question about the Mosquito Assassins? And for all of you who need to go, please go. Have a lovely evening.
But I do have one from Dorothy Dow, and she also has left the webinar, but it's a great question. She says, I find that these uncommon pollinators have a physical look with a metallic blue-yellow color on their abdomen. Not just these mosquitoes, I observe from other species like the red and black mason wasp with its colors are actually pollinators too.
Yes.
So those who don't necessarily pollinate as their main side job, they tend to be dull in color, maybe black. She'd like to know if you could confirm that with her.
So what she is saying is that pollinators in general, or insect pollinators in general, are more brightly colored, whereas insects that are not primarily pollinators are not. I can't confirm that.
Okay.
But it is an interesting observation. I'm going to have to look a little bit closer. There are plenty of mosquitoes that will take floral nectar, therefore considered secondary or lesser pollinators that are not brightly colored at all.
Okay.
In general, though, animals or insects that are more diurnal or day active are more brightly colored because they have to be seen by their potential mates.
Oh, that's interesting.
Some Culex, for instance, the Pipiens complex are just a dull brown. They mainly fly in the evening and at nighttime, dusk and nighttime. So they don't really need to be brightly colored because it's dark. So they need to find each other in other means.
Mm-hmm. Another question that someone posed, Anita, was about rudulus. And, what does rudulus mean? And I know that in Latin, rudulus means red or reddish. But
yes.
the Toxioronchides ridiculus, does that refer to the fact that they're kind of reddish as larvae, or is there any, is that, okay, I was wondering what the derivation was.
That's it. That's it. You got it. You pegged it.
Okay, great, thank you. Fantastic.
So for Toxioronchides rutilus, I have seen them in different shades of red and even purple. So they are different from other mosquito species in their exoskeleton construct, that they are quite pigmented. Where some of our culex species will be green, even Anopheles will be green or yellow often, just depending on their food intake, depending on what they eat. Whereas rutilus, the exoskeleton itself is pigmented and is a reddish color. But if they take in a lot of food that is, say, dark or almost dark black, then the larva will become much, much darker. If they take in food that has a bluish hue, then they will become almost purplish.
That's so interesting.
In different color. And then when they first eclose, I don't know if you saw that in the biology slides. I do have an image there of a microscopy image of a tox larva. It's about 10 minutes old, and it's completely transparent. Hasn't sclerotized. The head capsule is still soft, and it's completely see-through.
Those were beautiful pictures, and I also really enjoyed seeing the ovipositing of the Toxioronchides that you showed us too. That was pretty cool.
Yeah, that's a pretty cool video.
I had never seen that, so this was such a treat.
Thank you for inviting me.
Thank you again for our whole team and all the people that were